Hi everyone, I am Dr. Anisha, your microbiology faculty, and today I will be discussing about two very important hypersensitivity reactions that are hypersensitivity 3 and 4. Now, before we talk about hypersensitivity, we must know one thing, that for hypersensitivity to occur, there needs to be a prior exposure to an antigen, right? So, suppose a person has been exposed to a particular antigen, right? So, if the person, he shows or the body shows any exaggerated response, any injurious response, or any sort of inflammatory response, it is only then we consider it as hypersensitivity, right? So, when we talk about type 3 hypersensitivity, so we have devised 4 letters for each type of hypersensitivities, respectively type 1, 2, 3, 4. So, for type 3 hypersensitivity, C corresponds with type 3 hypersensitivity, right? So, there are two things that are going to happen in this particular type of reaction. One is immune complex or excessive formation of immune complexes, C for C, right? And another thing that happens in type 3 hypersensitivity, it is the activation of complement cascade. Activation of complement cascade occurs. Right? So, two C's, C for complexes and C for complement. Now, we know that for any hypersensitivity to occur, we require antigens for the antibodies to act upon, right? So, what are these antigens? What can these antigens be? These can be anything. These can be bacteria, these can be viruses, or these can be any DNAs, exogenous or endogenous antigens, which are acting as antigens, acting as foreign bodies, or they will be associated with immune responses or exaggerated immune responses, as we say. So, these antigens can be bacteria, viruses, or any endogenous DNAs. So what happens? So normally a lot of antigens, we are getting exposed to a lot of foreign bodies on daily basis, right? So not every foreign body, it leads to formation of larger immune complexes and leads to tissue destruction, right? So under normal circumstances, although antigens are getting entered, they're entering inside a body, and antigen-antibody complexes are forming normally also. But these antigen-antibody complexes, instead of causing tissue damage, they are getting phagocytosed. How? So once these antigen-antibody complexes form, so what is happening? So they are getting coated with complements. They are getting coated with complement proteins, complement components, right? And after getting coated with complement components, what are they getting? They are getting phagocytosed. They are getting phagocytosed by macrophages, right? They are getting phagocytosed by macrophages or monocytes. Or when these antigen-antibody complexes are small enough, they might attach to RBCs, antibody-antigen complexes. They attach to RBCs, and via circulation, they are ultimately taken to the graveyards. And what are the graveyards? The spleen and the liver, right? So via circulation, they are getting destroyed inside what? Inside spleen and liver, right? So this happens under normal circumstances. These complexes not always lead to formation of tissue destruction, right? So, but there are certain conditions when there is a longer duration of antigen exposure, a longer duration of antigen-antibody complex formation. So there are repeated exposures, repeated complex formations, which ultimately lead to the higher, or they lead to greater tissue destruction conditions, right? So what are these longer durations? These can be firstly chronic infections. Chronic infections when there is a lot of antigen which is getting presented inside our body. A lot of antibodies are formed for those particular antigens, and this cycle continues for a longer period of time. So when this longer duration occurs, a lot of tissue destruction is happening along, right? So one is chronic infection. Then if there are repeated infections, right? Then alongside there are prolonged duration, prolonged antibody reactions, immune responses occurring. And also in autoimmune conditions where our cells are being presented to the antigen presenting cells at times, or they are acting as antigens to be acted upon, right? So these cells are acting as antigens, and they are leading to a circle of tissue destruction, right? So in autoimmune conditions also, there is a prolonged duration of antigen-antibody complex formation, right? So what is the pathophysiology behind all of this? How is the tissue destruction occurring in type 3 hypersensitivity reaction, right? So we have an exogenous antigen. This is the antigen which enters inside our body, which our body in response, which leads to the activation of B cells, right? B cells get stimulated. They in turn, they stimulate the plasma cells to release antibodies, right? This is the whole mechanism that takes place whenever an antigen enters inside a body. So antigen-antibody interaction occurs, right? Antigen-antibody interaction occurs. Then these antigen-antibodies, they form complexes. Complexes are formed, right? And these antigen-antibody complexes, these get deposited. Now these antigen-antibody complexes, they are coated by complement. Now what does complement do? Now after getting coated by complement, the complement activates, right? So which pathway is getting activated? It is the classical complement pathway. Which is getting activated. You must remember this in type 3 hypersensitivity. Classical complement pathway is getting activated. So once it gets activated, what does it release? It releases certain anaphylatoxins, and along with anaphylatoxins, there is also release of chemoattractants, right? And what are these anaphylatoxins? These are C3A and C5A. So what are these anaphylatoxins going to do? These guys are going to release what? These lead to the mast cell degranulation. In general terms, this happens. Mast cell degranulation occurs. So on mast cell degranulation, ultimately what is happening? The end result is going to be tissue injury. And how is that tissue injury occurring? Because of increased permeability. Increased vessel permeability occurs. This is one thing. Mast cell degranulation, and ultimately the end point is again going to be tissue injury, destruction, right? So these anaphylotoxins, alongside, they also act as chemoattractants. And why do we say they act as chemoattractants? Because these guys are going to attract the neutrophils from the circulation to the site of the immune complex formation and the complement coating and everything, and the tissue destruction site, right? So these neutrophils are going to be recruited. Now what are these neutrophils doing? Neutrophils are going to release lytic enzymes, right? So now the ultimate goal of recruiting these cells, these inflammatory cells, is to get rid of those immune complexes which are getting deposited, right? But these neutrophils, these are not potent enough to kill those large, destroy those large immune complexes. So what it does, instead of getting those immune complexes destructed, these neutrophils, they release lots and lots of lytic enzymes. So instead of clearing those immune complexes, these neutrophils, they lead to excessive tissue destruction. So instead of acting as helpful cells, they lead to more and more tissue destruction. They lead to excessive tissue destruction, right? So neutrophils are also recruited, mast cells also degranulated, vessel permeability occurs, right? So ultimately what is happening? A lot of tissue destruction is happening. The next thing that occurs is there is the formation of platelet aggregates. So platelet aggregation occurs, right? So because of platelet aggregation, microthrombi are also getting formed. Microthrombi are formed, more tissue destruction, more area is getting involved, right? And after that, fibrous tissue is also getting formed. There is also fibrosis which is occurring. Fibrosis, platelet aggregation, right? This whole mechanism leads to a larger tissue destructive event, right? So all of these are occurring at the site of tissue destruction. So now we must know for type 3 hypersensitivity reaction that there are two reactions occurring in type 3 also. It is an immune complex mediated, we are well aware of, right? So what are these two types of type 3 hypersensitivity reaction? These can be localized reactions, or these can be systemic or generalized reactions. Right? Now, what actually decides whether it is going to be a local reaction or a systemic reaction? So what happens is, two scenarios occur. There are two scenarios which we will be discussing about. Firstly, see if there is antibody excess. if large number of antibodies are present and smaller amount of antigens are present, antibodies are larger in size, right? And if there is equivalence of antigen as well as antibody, right? If there is equivalence. Now what is occurring because of the larger size? So these antigen-antibody interaction leads to formation of larger immune complexes, which instead of moving into circulation and getting deposited in other sites, these immune complexes, they will get localized or deposited at site of inoculation or at a particular site where these antigen antibody complexes will get deposited and they will be coated with the complement, right? So this leads to a localized response when the antigen antibody complexes are large enough that they cannot travel to different sites, so they get deposited at the particular site and they cause the localized tissue destruction, erythema or induration, etc. Right? Now when there is, and these immune complexes, these large immune complexes, you must remember these are insoluble, right? So the other scenario is when there is antigen excess. So when antigens are in majority, so they will lead to formation of smaller immune complexes, right? So these smaller complexes are easier to travel from one side to another. So they travel via circulation to different sites, and they get deposited. And this is the scene when occurs a systemic response. So it is basically dependent on the size of immune complexes, whether there is antigen excess or antibody excess. So what is this localized immune response in type 3 hypersensitivity called? It is the Arthus reaction. So Arthus reaction, it is nothing but a localized tissue necrosis which occurs because of vasculitis because of local deposition of antigen antibody complexes, right? So in this image, can you appreciate this area, these red areas of induration as well as erythemas? These are nothing but localized tissue destruction which are caused by antigen antibody immune complex depositions, right? So this is Arthus reaction. And why do we call it Arthus in specific terms? Because Arthus in around 1903, he took an experimental animal, that is rabbit, which was previously immunized to a particular antigen, right? He had a lot of antibodies already present in his circulation because of previous immunization, right? So once this person, he injected the antigen again in that rabbit, it led to local tissue destructive necrosis or tissue destruction occurred. So it is with the experimental animal, with the all the local destructions or all these signs and symptoms, he named such reaction as Arthus reaction, right? So this is why we say that there is localized tissue response, all the erythema and everything that occurs. So now this Arthus reaction is not limited to skin only. It is not just a dermal condition. It can be occurring in lungs as well, right? So why do we call it a skin infection Arthus reaction? How can we see this in normal conditions in us humans? This can occur after an insect bite. After an insect bite, a lot of you might have experienced insect bites where you very well know there is a lot of erythema, induration, pain, redness, everything that occurs at the local site, right? So this can occur after an insect bite, or when there is repeated injections of a particular antigen. So when there is repeated exposure, so these repeated exposures lead to deposition of the complexes at the site of inoculation only, so leading to localized response. Now what happens in lungs? There is going to be an allergic reaction to a lot of antigens which we inhale, right? So there are two very important conditions which are asked in your exams related with type 3 hypersensitivity. These are farmer's lung and 𝗫𝗫𝗏𝗔𝗟 disease. What happens in farmer's lung? There is inhalation of actinomycetes from moldy hay. So the person develops allergic type of respiratory issue in such situation. So the immune complexes, they are getting deposited in the alveoli or respiratory passages, leading to disease manifestations of this condition, farmer's lung. So you must remember, it is the inhalation of actinomycetes from the moldy hay. And for 𝗫𝗫𝗏𝗔𝗟 disease, it is the inhalation of serum proteins from the dust which has been derived from the pigeon droppings, pigeon excreta droppings, leads to 𝗫𝗫𝗏𝗔𝗟 disease. Now for questions in your exams, you're asked what are the examples of localized type 3 hypersensitivity reactions. So in order to learn that, in order to memorize it, I have a mnemonic FIR. So it's a story that two guys are fighting and they are forming immune complexes, and a third person spots them and he immediately files an FIR. So what does this FIR stand for? F stands for two F's, farmer's lung and 𝗫𝗫𝗏𝗔𝗟 disease, right? I stand for insect bite, and R stands for repeated injections. So this will make you memorize the localized Arthus reactions, the immune complex deposition, and an FIR has been filed against those two guys. So what are the generalized or systemic type 3 reactions? So you must remember that these reactions occur in two phases, right? So firstly, there is formation of small size soluble antigen-antibody complexes. You must know that these small size antigen-antibody complexes are soluble in nature, right? And when we were discussing about the immune complexes, two scenarios were occurring when there was antibody excess. We spoke that there are large immune complexes which are getting formed, and these are insoluble in nature. Whereas these small complexes, these antigen excess complexes, these are always going to be soluble complexes, right? So now, there is deposition of these complexes. Firstly, formation of small immune complexes are occurring, right? And via circulation, these are getting transported to different sites and they are getting deposited at those sites. So what are these sites or these tissues where these deposits or complexes are getting deposited? These can be very well, most common, these can be blood vessels, because of which they will be responsible for vasculitis, right? Then they can get deposited in synovial membrane, which would be responsible for arthritis, different types of arthritis, right? And they can also get deposited in glomerular basement membrane. So these are the most common sites of deposition of immune complexes, right? So now the most favorite question from type 3 hypersensitivity, the examples of type 3 hypersensitivity. You will be given clinical scenarios and you will be asked which type of hypersensitivity this is, or you will be simply given 4 options of different diseases and you will be asked to mark out which hypersensitivity. Either ways it would be asked, right? So for that I have again a mnemonic. Remember when I said that 2 guys were fighting and the third guy, he spots those guys fighting and he immediately files an FIR, right? So on seeing this, that a person has filed an FIR, both of them decide that it's a Hindi mnemonic. So he says, "Pagal, stupid, let's just go and let's just run away from here," right? So this is the Hindi mnemonic which I have used. So P stands for firstly polyarthritis nodosa, right? Then next, A stands for arthritis, and this arthritis can be observed in hepatitis B, hepatitis C and dengue, right? Then G stands for post-streptococcal glomerulonephritis. And what is this post-streptococcal? Which infection it is? Streptococcus pyogenes, right? So it can be seen in post-streptococcal glomerulonephritis where there is deposition of immune complexes in the glomerular basement membrane, right? Then L stands for lepra reaction 2. Lepra reaction 2, right? Then for S, we have systemic lupus erythematosus, an autoimmune condition. Then we also have serum sickness, right? And with S, we also have subacute bacterial endocarditis. Subacute bacterial endocarditis. So with K, we have �'Τ fever, right? A stands for African trypanosomiasis. Then I, from this particular word, it stands for infectious mononucleosis. M, it stands for Plasmodium malariae leading to nephrotic syndrome, right? And R stands for rheumatoid arthritis. Now these are very important one-liners or maybe case-based questions which very frequently asked in your exams when we particularly discuss about types of hypersensitivity. So mostly these examples get asked in exams, right? So with this mnemonic, it gets very easy for you to remember. Now, serum sickness, it was a condition which used to occur in the past where in order to treat tetanus, Hawes anti-tetanus serum used to be injected into the patients, right? It was injected to treat tetanus. So this Hawes, because it was foreign, so it produced a lot of antibodies, a lot of immune reaction used to occur. So within 7 to 8 days only, a lot of immune complexes were getting formed, right? So patient developed erythema and there was vasculitis, then patient developed glomerulonephritis, then patient had fever, and all these conditions. Patients developed such symptoms, and once these immune complexes were cleared, these symptoms subsided, right? These immune complexes once cleared, so it was not a permanent situation, but it was a temporary condition which was considered as serum sickness, where a particular serum once injected, it led to a lot of inflammatory conditions, and further when these complexes got cleared from the circulation, the symptoms and all the associated conditions, they subsided, right? So it is not a scenario now, but serum sickness in itself is asked as a question, that it is an example of which type of hypersensitivity reaction. It is only because of the formation of immune complexes in the circulation. Now talking about type 4 hypersensitivity. So type 4 hypersensitivity, with D comes 4. So it is a type of delayed hypersensitivity reaction. And why do we call it delayed? Because there is a delay of around 48 to 72 hours for the symptoms to occur, right? And one important point that we have discussed, that A, B, C type of hypersensitivities or 1, 2, 3 hypersensitivities are humoral, type of hypersensitivity is humoral, right? And this is cell-mediated hypersensitivity or cell-mediated reaction. And what type of cells are associated with type 4 hypersensitivity? Again a question. It is the delayed type hypersensitivity cells, right? So these are responsible for type 4 hypersensitivity. Now, what is the mechanism of type 4 hypersensitivity reaction? We must know that this occurs in two phases. One is the sensitization phase and the other phase is effector phase, right? So what is occurring, what happens when we say we are talking about sensitization phase or effector phase? It is just that a particular antigen is saying hello to our body, right? And on subsequent exposure to that particular antigen, once it has said hello to the body, and again if it comes out, if a person doesn't like it, if our body doesn't like it, so it is going to lead to a lot of reactions which wants to get rid of that particular antigen. So it doesn't like it, it doesn't like its hello, so it gets angry, our body gets angry, and it releases a lot of mediators which is ultimately helping it get rid of that particular antigen. It doesn't like it, so it is trying to get rid of it, right? And instead of getting rid of it, along with a lot of anger destroys a lot of things, so does in effector phase a lot of tissue destruction is occurring. So in general terms, this is what we mean by sensitization and an effector phase, right? So sensitization phase, what happens in sensitization phase in type 4 hypersensitivity? So there is an initial phase of around 1 to 2 weeks which occurs after antigen exposure. So what exactly is happening in this particular phase? So suppose there is an antigen. So this is an antigen presenting cell in our body. So this particular antigen, say for suppose this is a bacteria. So our body is going to process this bacteria. It is going to take this antigen and it is going to process it and present it along with MHC molecules, right? It is going to present the antigen, right? So what happens next? Now there is going to be the interaction of the T helper cells, right? T cells occurs. Interaction of the antigen on the antigen presenting cell along with MHC molecules, interaction occurs with T cells, right? Now what happens after that? These T cells, they are going to release interleukin 2. Interleukin 2, which is a type of cytokine, and this interleukin 2 is going to lead to proliferation or differentiation of these T cells or helper T cells into delayed type hypersensitivity cells. So these T helper cells are majorly derived from Th1 cells, you must remember, in majority of cases. So now what is this interleukin 2 doing? This is asking this T helper cells that yes, I am getting released and I am going to lead to your proliferation, excessive proliferation of T helper, these delayed type hypersensitivity cells occur. So these T helper cells are getting differentiated into delayed type hypersensitivity cell. This is one major event which is occurring, right? So there is differentiation of delayed type hypersensitivity cell has occurred, right? So now this delayed cells, they have gotten sensitized to that particular antigen, right? So now what occurs in effector phase? So in effector phase, these delayed hypersensitivity cells, what they do, they are going to release a lot of these cytokines. A lot of cytokines get released, right? So one of these cytokine is interferon gamma, right? This is a major cytokine, a principal cytokine, which is released in type 3 hypersensitivity reaction. You must know. This is the principal cytokine, right? And the other cytokines which are getting released, these are TNF beta, right? Then there is the release of interleukin 3. Interleukin 2 has already released in the sensitization phase, right? Interleukin 3 is getting released. Then migration inhibitory factor is getting released. Then monocyte chemoattractant and activation factor is getting released. Then granulocyte, monocyte, colony stimulating factor, these are also getting released. So a lot of stuff is happening, a lot of cytokines are getting released. So what are the role of these cytokines? We have already spoken. Interleukin 2 is going to lead to proliferation of delayed type hypersensitivity cells 1, right? Then what is this migratory inhibitory factor doing? So one of the function is that, so once these cells are recruited, a lot of phagocytic cells are recruited, this migratory inhibitory factor is going to stop the migration of these inflammatory cells or macrophages from the site of tissue destruction to other cells. It is going to stop it. Please don't go from here. You stay here only and cause more and more tissue destruction, right? And these monocyte chemoattractant and activation factor, what is it doing? It is causing more and more, it leads to more and more attraction of these monocytes at the site of tissue destruction. So they are getting attracted at the tissue destruction site, right? Then local proliferation of local recruitment of monocytes is also occurring, right? And TNF beta, this is also, all of these are, what are doing? Ultimately they are causing recruitment of inflammatory cells. Right? So the major function is they are recruiting more and more inflammatory cells so that they form larger tissue destruction at a particular site. So what is this interferon gamma doing? We said it is a key cytokine. It acts as an alarm clock. And for what is this alarm clock set? It is set for the resting macrophage. So it is going to wake the resting macrophages that you have to do tissue destruction, right? So what is it doing? Initially, it is going to activate the resting macrophages. And how are they doing? By releasing certain cytokines, interferon gamma. Interferon gamma has already led to the activation of resting macrophage. Now this resting macrophage, it is getting activated, right? On activation, what happens? So there is going to be, firstly, in large number of MHC molecules are getting presented. Presentation of large number of MHC2 molecules occurs. Then large number of TNF beta receptors are getting presented, right? Lot of oxygen radicals are getting released, right? And nitric oxide, it is also getting released. So these are all acting as inflammatory things. So this activated macrophage is now ready to cause, it is very active, it is ready to cause a lot of tissue destruction. Tissue destruction. Right? So now we have said a lot of things are happening, sensitization phase has occurred, macrophages are getting activated. So what is the main role of these delayed hypersensitivity reaction? Does it always end up in, larger destructive events, or can it also provide a protective response? Right? So the main function of these cells is that it tries to protect the body against intracellular pathogens and other chemicals. But in doing so, because a lot of these intracellular pathogens are difficult to kill, so in order to kill those intracellular pathogens, they release lots and lots of lytic enzymes, lots and lots of cytokines are getting released. So because of that, large number of inflammatory events are occurring, right? Inflammatory events occur because instead of protecting that, a lot of inflammatory reaction occurs. And because these are large, these intracellular pathogens, they at times they escape those macrophage killing or the phagocytic actions. They escape and they ultimately lead to larger granulomas, larger, necrosis, necrotic sites, necrotic areas in particular sites, right? So these, after getting escaped, they formed larger necrotic tissues. Necrotic tissues, right? So now we have spoken that there are intracellular pathogens which are majorly involved in delayed type hypersensitivity reaction, and also certain chemicals which induce the delayed type hypersensitivity. So what are these intracellular pathogens? Again, very, very, very important questions. So let's learn these with the help of another mnemonic, which is her most loved internal mycology TB lab at PHC and CHC. So H stands for herpes simplex virus. M stands for measles virus. V stands for variola virus. Internal stands for intracellular. Then M stands for Mycobacterium leprae, right? TB stands, of course, for Mycobacterium tuberculosis. L stands for Listeria monocytogenes. A and B, they stand for Brucella abortus. And PHC stands for, P stands for Pneumocystis 𝗲𝗏𝗃𝗃. H stands for Histoplasma capsulatum. Then another C stands for Candida albicans, and the last C stands for Cryptococcus neoformans. a very important section from the hypersensitivity 4. So you must remember these intracellular pathogens. Now we said that there are other components also which lead to type 4 hypersensitivity reaction. So these are nothing but certain compounds which are leading to contact dermatitis, right? So what is happening in contact dermatitis? Let us see. So firstly, let us know what are these chemicals. Nick, we have a mnemonic. Nick loves his IV poisons at picnics, right? So N stands for nickel, IV stands for poison ivy, and poison stands for poison oak, and picnic stands for pickerel chloride. Now what are these compounds? What are these chemicals? These chemicals, these are haptens, these are incomplete proteins. So what they do, they complex with skin proteins. And these skin proteins, they are taken by skin macrophages or Langerhans cells, which are presented to helper T cells. And after getting presented, they are again getting differentiated into delayed type hypersensitivity cells, right? And delayed type hypersensitivity cells leads to what? This leads to local tissue. These leads to local destruction, local granulomas, which form local tissue destructive sites. So this is the pathology behind contact dermatitis and how are these acting as certain hypersensitivity events, right? So now there are also other conditions associated with type 4 hypersensitivity which can lead to such delayed reactions, right? So these other conditions, again I have a mnemonic, a fun mnemonic rather. It is get perfect hash browns and croissant from Delhi Mall. So G stands for graft versus host reactions. Graft versus host disease, right? Then P stands for peripheral neuropathies. Then H hash stands for Hashimoto's thyroiditis. Right? Then C stands for Crohn's disease. Right? D stands for diabetes mellitus type 1, right? And M stands for multiple sclerosis, and L stands for lepra reaction type 1. So in type 3 hypersensitivity we read it is the lepra reaction type 2 which is occurring, which is quite the opposite case with type 4 hypersensitivity where lepra reaction type 1 is occurring. So with this mnemonic we have covered almost everything which gets asked for examples from these two types of hypersensitivities. So now moving forward to the pathology behind the granuloma formation which occurs in these conditions. A lot of granulomas are forming. We are well aware of granulomas in TB. Everybody knows that granulomas form in TB, right? So what are these forming? How are these forming? So there is continuous delayed type of hypersensitivity reaction which is occurring, right? So with time, these TH cells, they get replaced by macrophages in around 2 to 3 weeks, right? So there is excessive proliferation of these T cells, right? And then, can you appreciate this area? These are nothing but intracellular bacteria. And can you see these? These are nothing but these flat cells. Can you see these flat cells? These are nothing but the epithelioid cells, right? These are flattened eosinophilic cells. And then when these epithelioid cells, they fuse together, they lead to formation of multinucleated giant cells. This whole area, this whole amalgamation of multiple types of cells lead to formation of granulomas, and this particular formation is the granuloma, right? So inside we have multinucleated giant cells which are lined by epithelioid cells, right? Epithelioid cells. Then we have T cells, right? Outer covering of T lymphocytes is present. And then finally, outside these granulomas is the formation of fibrous tissue as well. Fibrous tissue and vascularization occurs. So if asked, this is what granulomas look like. So what are these granulomas? In what conditions can we spot these granulomas? So these can be found in tuberculosis very well. These can be found in leprosy. These can be found in sarcoidosis, right? So these are the conditions where we can see these multiple granulomas, right? So all these are asked as questions. So now moving on to the skin test for delayed type of hypersensitivity reaction. Again, a question which is very, very favorite among examiners. I have a mnemonic: forensic medicine and toxicology lab for the skin test, right? So F stands for free test, which is done for lymphogranuloma venerum. Then M stands for Mantoux test, which is done for TB, tuberculosis, right? Also known as tuberculin test. No, M stands for, along with Mantoux, it stands for, I have considered it as Montenegro test. Test which is done for leishmaniasis, right? Then L stands for lepromin test, which is done for leprosy, right? So these are all the types of tests which are done for delayed type of hypersensitivity. Now another question which gets asked is this tuberculin test. So what happens in tuberculin test? We have already sensitized individuals who have had prior exposure to TB bacilli. So what we do, we inject purified tuberculin antigen, right? We inject in certain sensitized individuals, and after 72 hours, what we look for induration and erythema, and if that occurs, then the person is said to be hypersensitive to tubercle bacilli. So these were all the important points and high yield sections from hypersensitivity reactions, along with very important examples and pathophysiology. So this was all about hypersensitivity 3 and 4. Thank you.